All right, hi everyone. Thanks so much for coming. We're really excited to have Dr. Rita Caldwell here today. My name is Amalia Almada, and on behalf of the Planetary Health Alliance, I'd like to extend a warm welcome to all of you and to our special guest. Before introducing Dr. Caldwell, I wanted to share a little information about the Planetary Health Alliance and who we are. We are a consortium of universities, NGOs, and government agencies based at Harvard Center for the Environment in partnership with the School of Public Health. The Alliance is designed to support the growth of a research field of planetary health, which we define as an interdisciplinary research field at the intersection of global environmental change and human health. So I would like to encourage you to visit our website, check us out on social media, or sign up on the sheets that we're outside and may be passing around to learn more about the programs we've been holding globally and here on Harvard's campus, including visits like those of Dr. Caldwell. We also invite you to attend the inaugural Planetary Health Annual Meeting that's going to be happening here at Harvard at the end of April. We're organizing this meeting along with the Ecological Society of America, the American Geophysical Union, and the Lancet to really start connecting this diverse group of students, researchers, and policymakers who are committed to understanding and communicating the human health impacts of global environmental change. So now to introduce Dr. Rita Caldwell. Throughout her career, Dr. Caldwell has been committed to supporting basic research and applying its findings to real-world problems. Her research interests are focused on global infectious diseases, water, and health, and she is currently developing an international network to address emerging infectious diseases and water issues, including safe drinking water for both the developed and developing world. Dr. Caldwell has served as the president of the American Association for the Advancement of Science, the American Institute of Biological Sciences, and the American Society of Microbiologists. From 1998 to 2004, she served as the first female director at the National Science Foundation. Dr. Caldwell now serves as Distinguished University Professor, both at Johns Hopkins University, Bloomberg School of Public Health, and at the University of Maryland Institute for Advanced Computer Studies. Dr. Caldwell is beloved by her family and her extended family of scientists and policymakers that have had the privilege to work alongside her and receive her generous mentorship, myself included, and others in the room as well. So it is an honor to have her here with us today. Without further ado, Dr. Rita Caldwell.
Thank you very much, Amalia. It's a pleasure to be here amongst friends and colleagues with whom I've worked. I'm going to probably use too many slides. My husband accuses me of giving subliminal lectures. The slides go by so fast. But there's so much that I want to tell you, and it's such fun to have an opportunity to share with you some of the things that we've been able to do. I like the Hippocrates quote because it really does describe, I think, what the Planetary Sciences Group is trying to do and the interest that I and my students have had, and that's just to understand what happens in the environment that influences the health of individuals. And in fact, it should, I think, influence the practice of medicine as we move into the changes that occur with our climate as we move into the later years of the 21st century. I'm going to focus on water because, in fact, that is what I have been studying for a long period of time, but more to the point that when you discuss diarrheal diseases, we're really talking about over 1 billion cases per year and almost 2 million deaths that occur, particularly children under the age of 5 and the elderly. When you sum up all of the diseases, the infections, and the deaths that occur just from diseases transmitted by water that's unsafe, it gets to be astronomical. I will focus on cholera. It's I think, an ideal model for the interaction of humans and the environment, and particularly since it's a disease that has historical perspective and a direct correlation with the environment. It serves also as a model, as I will show, for prediction using environmental parameters. The Benial Delta has been known as the home of cholera. I think this is rather unfair because, in fact, I think just over 100 years ago, on the east coast of the United States, including Boston, Washington, particularly Washington, when it was believed, we didn't yet quite have the germ theory of disease, the consideration was that bad air, miasma, was the cause of infectious disease. And Washington, D.C. certainly was the home of malaria, yellow fever, cholera, a variety of diseases. It was known as a miasmic swamp, but I think it still is for different reasons. The cholera story really goes to the environment and the relationship of copepods of the bacterium, which we can culture easily in the laboratory using selected media. But copepods have been shown by the work that we have done and others to carry a lot of cholera bacteria, as many as 50,000 colony-forming units of cholera when you plate it out on a selected medium. The bacteria have been shown by Richard Cash here at Harvard in the School of Public Health and colleagues to be a dose-dependent disease. In other words, if you ingest a few hundred of the cells, you'll probably have a mild case of diarrhea. You won't feel so good, but you'll recover. But if you ingest 1 million cells per mil and in a slug of water, in a glass of water, then you'll end up very likely with the typical dehydrating, severe form of cholera that can result in death if the fluid's lost and not replaced. One of the other discoveries that we made, aside from the fact that it's a naturally occurring bacterium in the water, 20% of the isolates we found are luminescent, and also all of the Vibrios have a chitin digesting enzyme, which means that their association with plankton, and particularly crustaceans like shrimp and crabs, is that once their host has died, they simply convert the chitin to carbon in the cycling in nature. But one of the phenomena that we were able to characterize is that of the non-culturability, where having built a monoclonal antibody that allowed us to see the organisms and count them, the numbers were much higher than that which we could culture. And that difference, of course, are the organisms that are viable but they're not culturable. In fact, if you produced a whole non-culturable set of Vibrios by simply putting a culture in the refrigerator, after a few days you won't be able to culture them, but you'll be able to find that they're still viable by adding substrate, particularly a labeled substrate that allows the cells to elongate, but they won't divide, but they will respire because you can pick up the radio-labeled CO2. So this phenomenon, which CDC referred to as Caldwell's ghosts, now there are about 1,000 publications on the non-culturable phenomenon that occurs in the gram-negatives, and I had the satisfaction a couple of years ago of reviewing a paper for PNAS from CDC on the molecular basis of non-culturability, so I think it's more or less accepted these days. So summarizing that early aspect of our work, clearly the bacteria are widely distributed in the environment, associated with a variety of organisms in the environment, and certainly in those areas of the world where sanitation is not very effective, person-to-person transmission occurs, but basically the source is in the environment. So that, I think those arguments are now well past, and the notion that the bacteria are part of the natural flora of the environment, autochthonous, to use a fancy word, native to the environment, these Vibrio cholerae, and also the fact that they do go into a dormant stage because their host, for example, the plankton, go into a diapause in the winter months, burrowing in the sediment and then reappearing in the spring to cast their eggs out into the water column. So that non-couchability is simply an evolutionary component of the life cycle of the Vibrios in accordance with their relationship with their natural host. Now, let me discuss a little bit about the environmental parameters that we've spent a good deal of time measuring, particularly in the Bay of Bengal. It occurred to us that in this relationship of plankton, we could probably measure using satellite sensors, because first you have, when spring months, the warming of the water, increased sunlight, and the appearance of phytoplankton, which you can measure. The chlorophyll can be measured by satellite sensors. And so if you do a calculation of about 6 to 8 weeks later, you would have the bloom of the zooplankton, which graze on the phytoplankton, which cannot be measured directly by satellite sensors, or at least I think it can in some of the work that's being done at Columbia Lamont Doherty Laboratory. I understand that some of the calculations can be done and automated so that you can measure zooplankton directly. But in any case, using chlorophyll as a surrogate measure and then doing a calculation of the time frame. And actually, with the early work, we hypothesized this as a way of measuring cholera and the relationship of the host back in 1996. And then we published a paper in PNAS using collaborative work measuring chlorophyll and sea surface temperature with NASA, the team in California. And then we've been able to use some of the more sophisticated satellites that I'll be discussing to measure even more taking more sophisticated measurements by satellite sensors. The fact is that cholera is globally distributed. The bacteria are in the environment in tropical and subtropical regions. The 1991-92 epidemic in Latin America was kind of an eye-opener, because that we could correlate with the El Nino of the 1991-92 years. We've still considered, however, that where the original description of cholera was done in the Bay of Bengal in Bangladesh, the work that we have done shows a correlation of the Himalayas, the Gangetic Delta River system, and we've done a great deal more work characterizing the interactions, particularly in Bangladesh, in Matlab, in the villages outside of Dhaka city. The red areas are the hotspots of cholera. The blue are the ponds that are excavated, and the villagers build their houses on the earth that has been excavated, and then they work around the ponds. Now, the relationship is clearly with the sea surface temperature, the cyclical warming and cooling that goes with the spring and fall seasons of the year. And we've also found correlations with sea surface height, because, in fact, the tidal effects into the river, bringing the plankton of the coastal regions up into the river system, that relationship, I think, is also significant, and we've been able to correlate sea surface height along with sea surface temperature. But fundamentally, the relationship is with the plankton populations that are seasonal, spring and fall, and they correlate very nicely. Well, I say nicely theoretically, not for the individuals who live in Bangladesh, but they do correlate with the seasonal peaks in cholera in the spring and again in the fall. So we tied that relationship in a very early publication back in 2000. That was hard to think, 17 years ago. That seems like time goes by so quickly. But in any case, doing that correction or calculation of the time of the peak in the chlorophyll to the increase in the zooplankton, by making that so-called correction, we were able to show a very dramatic relationship of sea surface temperature in the Bay of Bengal, just off the coast of the Sundarbans in Bangladesh, with actual cases of cholera in Matlab. So that correlation was, in fact, very dramatic, and the seasonality could be then accounted for by the phenomenon of temperature, salinity, the plankton populations, and the relationships have been subsequently, I think, confirmed. We've improved the models. This is a sort of a second iteration with Guillaume Constantin de Magny, one of my postdocs, who is now doing work in Africa, in Mozambique, and working also with colleagues in Senegal. But with this improved model, we were able to be even more precise, to be able to correlate for Calcutta with every milligram per cubic meter increase in chlorophyll measured by satellite sensor. We could account for a 33% increase in the numbers of cases of cholera in Calcutta. And rainfall is another important parameter. So with every millimeter increase per day of rainfall, we could account for about 7% increase in cholera cases. And similarly with MATLAB in Bangladesh. We've subsequently developed even more sophisticated models that allow us to do some predictive capacity, particularly in Africa, being able to predict some of the outbreaks of cholera several weeks in advance of the actual outbreaks according to the environmental parameters. Now, there's also the macro scale phenomena that we've looked at, but we now know clearly that rainfall, flooding, the river height, because in the inland areas of Bangladesh, there's a single burst of cholera, and that's more or less related to the drop in the depth of the river, so that you have greater turbulence flow with mixture of the sediment in the water column, and that relationship then can be correlated with the cholera cases. Sea surface temperature I've already discussed, and salinity is a major factor, along with these, obviously, contamination and the direct person-to-person increase and enhancement of cases where sanitation is lacking. So with these macro scale processes, along with let's say you have an earthquake or some sort of social disruption, in the case of some of the gathering in India and in Bangladesh for festivals and religious observances, if that is concurrent with some sort of heavy rainfall from a hurricane or a monsoon, along with perhaps an earthquake, that kind of disruption, along with the pathogens themselves responding to increased temperature, and also the relationship of the immunity of the population that's being affected. All of these then can be built into a predictive model development. We've been able to describe cholera much more effective, and in fact in the audience is Dr. Akanda, who did his PhD at Tufts University along with Dr. Anta Judula, with whom I've been continuing to collaborate with both of them. And we've been able to characterize the cholera outbreaks much more precisely in terms of the peaks associated with flooding and with the intrusion into the river system as another major factor that allows us, along with the more sophisticated satellite measurements, to really begin to define the outbreaks with very high precision and capability for prediction. Let me take you back a bit historically, because that's also gives us some insight into this relationship of cholera in the environment. And that is, along with Dr. Elizabeth Whitcomb, a very interesting person who did a PhD in India on the hydrography of that country, and then subsequently went back to New Zealand to earn her medical degree, and then ended up practicing for many years, running a neurological hospital in London, a very, very talented individual. But she made the discovery that in the Natural History Museum in London, there are the maps that were maintained by the colonial Brits when they were in India. And they were meticulous, as the British tend to be, in actually recording every single death from cholera. They called it the diarrheal infection and the fevers, which were malaria. So we have the records going back to 1873 to the present. And in particular, that allowed us then to compare the epidemics that occurred along the coast, the endemic cholera that occurred year after year after year, with the epidemic cholera that occurred inland, that we could correlate with greater precision some of the environmental effects. And here we have gathered all the data and transferred it from those large maps, several feet long and wide, where they had meticulously put each of the deaths that occurred in each year, map that fold back one after another. All of this has been transferred to the computer. So that has allowed us to determine with great precision that when there is an increase in the air temperature, then we could correlate that with the outbreaks of cholera, so that if there's an increase for several weeks prior to this sudden massive outbreak of cholera, we could refine the temperature relationship. And this allowed us then to develop the model where air temperature above average for the 2 previous months, which pretty much allows growth of the bacteria in the river system, and then rainfall above average, which allows flooding, and then a mixing of the sediment into the water column. And then if there is not a very good sanitary system, as might occur with breakage that occurs with an earthquake, then you would have a very, very high risk of cholera. Let me turn, and I'll get back to that in a moment, but let me turn to another bit of evidence that I think is really quite compelling. And that is that in collaboration with a team at University of Genoa in Italy and a group of genomicists in Germany, and then also with oceanographers at in England, we were able to do a study because at Southampton, at the oceanography unit there, they had been collecting plankton for the last 40 years with extremely good insight that these would be invaluable data. So they provided what they called plankton tows of opportunity by giving plankton tows to cruising ships, folks that had yachts, ferries going back and forth between France and England, and cruising, the large cruising ships. They would provide these plankton nets, and then they would just simply collect, do tows, and then send the plankton to Southampton. So their plankton there, resident, that had been captured or collected 40 years ago, 30 years ago, 20 years ago, et cetera, et cetera. So Dr. Pruzzo, Carla Pruzzo, a wonderful scientist to collaborate with, she, using some of the probes that we had developed for Vibrios and some that they had developed as well, gene probes, went into the Southampton along with the German colleagues who are the molecular biologists and extracted DNA from the plankton samples for each of these years, and then purified the DNA and probed it. And quantitatively, with samples collected where there had been measured increase in sea surface temperature in the North Atlantic, then at these sites, off Nova Scotia, Newfoundland, in the central North Atlantic, off the Iceland coast and so forth, collected again, took those samples that had been collected in those areas and then recent collections as well, and we were able to demonstrate then the yellow are the Vibrio cholerae, the green are Vibrio parahemolyticus, and the pink are Vibrio vulnificus. Vulnificus is a particularly nasty bacterium. If you get a cut and it gets infected, you really have to be treated quickly because you can die within 24 or 48 hours if it goes systemic. It liquefies the internal organs and it causes terrible infections, and even if it doesn't go systemic, you often will lose a limb. I gave a talk in Chesapeake Bay a few years ago on the pathogenic Vibrio vulnificus, and in the audience was a victim who had lost a leg, and then a family member who described the death of one of their family members from the Vibrio vulnificus. So in any case, let me just point out that what we have observed is that there's been an increase in these Vibrios in these different sites associated with that warming of the sea surface. And when you put this together with the epidemiological recorded infections with Vibrio, not so much Vibrio cholerae in Europe, but increases in Vibrio parahemolyticus, which you get from ingesting fish and shellfish, particularly those who like to eat raw oysters and don't cook crabs very well, or fish. And so there is an increase in the infections off the Atlantic coast and in Europe associated with the increase in the Vibrios associated with the plankton, which we were able to determine because they were stored and we could extract the DNA. So that again is another indication, I think in this case, probably the first demonstration of an actual human health effect associated with climate change, in this case an infectious disease. Now, could we have predicted the outbreak in Haiti? Now, this is very controversial, and I'm a controversial figure, but I don't mind presenting my data. Argue from the data, you can interpret it as you will. But in any case, we have done studies of the outbreak that occurred in Haiti. The interesting point is that we were able to get samples from Haiti within about 2 weeks of that outbreak, which occurred in 2010, a year in which they had the worst earthquake, I think a Richter scale of 7 or 8. It was a really fierce earthquake. And it's a limestone geology. And with the earthquake, a good deal of grinding, and I've worked and talked with geologists who say that what will happen is that the pH of the water would go up with all of that limestone deposition, and we've measured the pH, and indeed the river in Haiti is now close to 9. And to isolate Vibrio cholerae, you use a pH of about 8.6 to 8.7, and you used elevated temperature. So in Haiti, where the initial outbreak was supposedly to have occurred, almost simultaneously with that outbreak, these cases were occurring all along the coast within a matter of a few weeks. And Dr. Akanda, who's in the audience, and Dr. Jutla, along with Alam at Tufts, did calculations, and actually to reach from here along the Artemibi River down into the coast would have taken weeks, whereas this was occurring rather almost simultaneously. But more to the point is that we were able to get samples from cholera victims, stool samples, and were able to do isolations by working closely with Dr. Prosper, a physician, a Haitian physician trained in the U.S., and with a technician who happened to be working with a church group from Maryland, and the wife of one of my students was actually in Haiti, so we were able to collect samples rather directly and do analyses. Now, Dr. Jutla did a retrospective analysis and was able to pull together the air temperature in Haiti for the last 50 years. It was the hottest summer in 50 years in 2010. And a hurricane skirted the coast of Haiti just at the end of October, beginning of November, when the outbreak began. And it was the heaviest rainfall in 50 years. So high temperature, heavy rainfall, and the elevated pH of the river meant that there was a selective factor probably occurring in the rivers of Haiti where that the earthquake had occurred, so the conditions were optimal. We were able to study the isolates from 81 stool samples, and we found that half of them had Vibrio cholerae O1, the epidemic serotype, and 21 of the samples did not have any of the epidemic Vibrio cholerae, but had the local, non-toxigenic, non-O1 Vibrio cholerae. We have subsequently published a paper of an outbreak that occurred a couple of years later off the coast of Florida of cholera caused by non-O1, non-toxigenic Vibrio cholerae. So the non-O1, non-toxigenic do and can cause cholera. Not always, but can. So here we had 21 of the samples did not have the epidemic strain. They did carry the non-O1s, and some of them also carried Anaeromonas, just Anaeromonas bacterium, which can cause a cholera-like disease. Now, along with Dr. Mekalanos at Harvard and my team at Maryland and Tiger, in 2000 we published the first sequence of Vibrio cholerae and showed definitively that there are two chromosomes, which had been hypothesized by one of my former students, Jim Caper, using traditional microbial genetics, and we were able to confirm that in fact this is not a plasmid, but it is a chromosome because there are many metabolic genes. We have subsequently sequenced well over 400 Vibrio cholerae now. Here are some that we published back in I think that was in 2009, a few years ago. These are from all over the world, different Vibrio cholerae. And just if you could just stretch out the chromosome, the large chromosome and the small chromosome, and then match all the others up against it, we found that none of the Vibrios are absolutely identical one to another. There are some base pair differences. So we then have analyzed all of the Vibrio cholerae that we isolated from Haiti, and we found that in fact they formed a cluster separable from all of the others that we had analyzed. And then when we did principal component analysis, there was one of the strains from other epidemics from Asia and in that Haitian cloud, but that strain turned out to be from Zambia. So I think from my perspective, this is a much more complicated situation, and it certainly was fed by the environmental parameters, the hottest summer in 50 years, the heaviest rainfall in 50 years, the earthquake that caused terrible clustering of individuals in refugee camps, hence the bad sanitation. So all of this worked together to create the horrendous outbreak, there's no question about it, the worst outbreak in recent years in that country. There are many other parameters that we measured, differences according to polymorphisms and different mutations in different genes that characterize the Haitian organism. Now let me take you to another set of work that we're doing that I think are very exciting, and it ties all in. It's the molecular, greater detail on the molecular approaches. We were able to do a study and develop a technique for being able to identify everything, all microorganisms in samples, using extraction of DNA, whether it's a food sample, a water sample, seawater sample, urine, blood, sputum, stool, it doesn't matter. You extract from the sample all of the DNA. The extracted DNA then is sequenced, and it doesn't matter what sequencer. It can be PacBio, it can be Illumina, any one of the Illumina, it can be Ion Torrent 454, it doesn't matter. And then the raw sequence reads are matched up against the exquisite libraries that we've developed over the last 8 years. We now have 65,000 highly curated microorganisms, bacteria, viruses, fungus, and parasites. The matching is done within minutes using a Bayesian probabilistic approach, and it allows us then, with a probabilistic matching, to identify the bacteria down to species and strain. We also identify viruses, fungus, and parasites. And then we match that against the library that we've developed of antibiotic resistance genes, virulence genes, metabolic pathway genes, and that allows this then to characterize the presence not only of the species and subspecies and strains, but also the presence, the carriage of antibiotic resistance, pathogenicity traits, metabolic pathways. So let me describe a study that we have just completed, getting ready to publish, done in collaboration with a great team at the National Institutes of Cholera and Enteric Diseases, NICED, in Calcutta. The study that we did was to look at 74 samples, or individual stool samples. 20 were from Indians who did not, Calcutta inhabitants, either family members or from the Calcutta community, that did not have evidence of disease. They were free of cholera or any symptomatic diarrheal disease. And then we had 28 stool samples from those from which they could identify the presence of Vibrio cholerae. Another 26 that were ill, because this is a diarrheal disease hospital. They had diarrheal disease, very typical of cholera, but they could not isolate Vibrio cholerae, but they were sick nevertheless. And then we downloaded 20 individual samples from the human microbiome study done at NIH, from which we could draw the data and be able to compare Western, Northern European gut flora from the Calcutta Indian flora. So let me show you the results. They're really quite fascinating, I think. This is the very typical summarized data, and there was insufficient variation to say that individuals of those that had cholera, Vibrio cholera, varied. This was pretty much a uniform pattern. So the pink area are pathogens. The important point I want to make is that what the Calcutta team did was from the population that in Calcutta, from which they were able to get stool samples, they extracted the DNA, sent it to us blinded, and then they proceeded to do 26 tests, viruses, bacteria, and parasites, using the classical culture techniques. Now, they were surprised to find that when they did all those tests, and we were able also to show that when you had came in with a diarrheal disease looking like cholera, you usually had 4 to 10 pathogens. You did not just have Vibrio cholerae. So I think that we now need to move from the Koch's postulates of assuming there is a pathogen to the 21st century approach that there is interaction amongst pathogens, and what gets you to the hospital very likely may be polymicrobial infections. In fact, it's been demonstrated by others working in Calcutta that when Giardia is present, Vibrio cholerae produces much more toxin. It's more toxigenic. So there's interactions amongst these. In any case, let me explain these data. So indeed, we were able to isolate detect Vibrio cholerae, show its presence along with other pathogens like Campylobacter, Helicobacter pylori, and E. coli. When they could not identify and Vibrio cholerae, actually there seemed to be even more pathogens, and we were able to demonstrate they were mostly the E. coli superfamily, enteropathogenic E. coli, and a mix of them. It wasn't just enterohemorrhagic or enterotoxigenic or whatever. It was just a mix of all of the pathogenic type of E. coli. The other interesting phenomenon is that in the healthy control of the Indian population, they carry larger numbers of population that would probably make us ill. We Westerners, the pink is a very thin sliver, as you can see. But in the Indian population, the blue and the light blue are the kinds of bacteria you would get from ingesting probiotics, yogurt, buttermilk, the kinds of protective bacteria. We don't eat so much, but then we don't have so many pathogens in the Western population. But I think we can see this is so consistent that this becomes a bioforensic tool. So if someone were to be in the U.S. and to have said that they were native-born and had not been to India, and you did an analysis of their gut flora and they had just arrived, you would be able to detect that because the gut flora is very, very different. Now, we're able to identify the pathogens, and again, just another depicting, because here's listing of all the species, and these are the species that are particularly pathogenic, and you can, and going horizontally, vertically, these are the human microbiome, the Western gut, and you can see it is much less of the pathogens, as you can see in the known disease etiology and the unknown etiology, which are mostly the enterics. Not only are we able to characterize the superfamily of E. coli, many of the E. coli, but each one of them, we can characterize their accessory genes, and we're able to describe their functional characteristics so that we're able to show, first of all, they carry a whole lot more antibiotic resistance genes. So the asymptomatic control Indian population, because antibiotics you can buy from street vendors. You don't need a prescription. You can buy them in shops or even on the street. So the promiscuous use of antibiotics leads to the carriage of a great deal of antibiotic resistance in the normal population. Where the etiology is known, again, there's still a lot of resistance, which makes it difficult to treat. The beta-lactamases, the tetracycline, macrolides, and so forth. And the unknown etiology, even more intense antibiotic resistance, probably because they were treating themselves before they went to the hospital. So anyway, the other interesting aspect is that when we assemble the metabolic pathway genes, we find that carbohydrate metabolism is very dominant in the Indian population. And if you travel to India, rice at every meal, so it'd be expected to have a gut flora that's metabolically carbohydrate active. So a quick summary, the microbial communities of the Indian population is different from those of the Western European descent. There's a greater tolerance for pathogens in the Indian population, and multiple pathogens readily identified from the diseased patients. And the functional analysis, I think, is particularly interesting. Now, let me very quickly, I know I'm running out of time, but I want to make two other important points. One is that we are able to identify strain differences. This is really critical because I will describe now a study we've just completed, was published in PNAS a year ago, where we were able to show that two strains, different strains of the same species, worked together to cause the disease. A sad tale of a young 16-year-old woman who built herself a zip line. She was having a great time on the zip line. It broke. She fell in the water, gashed her leg, developed necrotizing cellular fasciitis. Time was spent looking for streptococcus because that's what usually is the causative agent. Unsuccessful, but they finally were able to isolate four isolates of Vibrio cholerae. When they were sequenced, 3 were clonal. 1 was quite different. To understand how these worked, because that victim, the infection went systemic. She had both legs amputated to the knee, and then eventually both arms in the vain attempt to try to halt the spread of the disease. She eventually died. Now, when we did the experiments to try to figure out what was going on, all four, this was done with Dr. Chopra at the University of Texas, Austin. He had a mice colony. He did the experiments where he injected into mice each of the four strains, the blue, red, green, and the lighter blue, or purple. And these were injected. We could pick them up, identify them. When we extract the tissue from the muscle injection site, extract the DNA, we could find all four. But when we analyzed the liver and spleen, we only found one strain. And so to prove that this was the case, we did classical genetic engineering. We introduced the luminescent gene and genes and also kanamycin resistant, repeated the experiment, and the only strain that went through to the liver and the pancreas was luminescent and kanamycin resistant. So our technique works by classical techniques. Of course, this technique takes weeks. Ours takes 5 minutes once you've extracted the DNA from the tissue. And we were able to show that really what was happening was that one of the strains, those three clonal similar strains, they carried a toxin, and they would damage the tissue, which allowed the other strain to migrate through to the liver and spleen. So here is where it's important to understand strains of an infectious agent, not just the species. And we've known this commercially because lactobacillus, one strain is used of a given species is used to make cheese, another strain is used to make wine, and a third is used for yogurt. So it's commercially we've known that strain differences are important, but now medically strain differences are quite critical. It is the toxin, the XOA, is a very similar toxin to that carried by Pseudomonas aeruginosa, and it is present in this Aeromonas species that cause it. Now, the other quick example is a study done with the University of Virginia where they were having antibiotic resistance being transferred to patients who were infected with other kinds of organisms, and it was creating a really difficult problem for them. And in tracking it down, it seemed most likely that it was the sinks that were the source of the infection. So we received some of the biofilm and extracted the DNA, did the tracking, and we were able, with this technique, to be able to show the distribution of the genes to the infective agents of patients in different hospital rooms. So we could actually track patient by patient using this technique, and we're able to demonstrate that it was actually plasmid transmitted antibiotic resistance that transferred from the bacteria in the biofilm in the sink through aerosol into the patient's rooms in the hospital. So again, polymicrobial infections are important. Being able to identify to the strain level is critical. And my last example is that we now are doing studies with water treatment in Orange County, California, because if you go to Los Angeles and you drink the tap water, it has gone through a lot of kidneys. It's sewage water that is reclaimed, but it's safe, very safe. So the sewage plant, solids piped out to sea, the water goes to the drinking water plant. It is then treated very, very well by microfiltration, reverse osmosis, ultraviolet, decarbonators, lime, chlorination, and then it is sprinkled over a wetlands, it trickles down for 3 months, drawn off, and that's the drinking water. That last step is very expensive, and the end point here, without having to spread it out on wetlands, really is drinkable. And so we've been able to work with the Orange County and looked at the Q1 represents the incoming water, and the microfiltration, biofilm, and then the reverse osmosis. We could pick up parasites in the incoming water, a few of the, not necessarily pathogenic, but some of the protozoa in the first stage, but once it's gone through reverse osmosis, no parasites. And then looking again at the water coming in, lots of pathogens, lots of bacteria, very diverse in the water coming in from the sewage plant to the drinking water plant. By the time it goes through the microfiltration, the diversity has dropped. There's still some pathogens. By the time it goes through reverse osmosis, it's almost only the typical water bacteria that ends up, in any case, in the water here or anywhere, because the tap water is not sterile, but it's safe. Now, with respect to viruses, all of the DNA viruses are removed, and the few animal viruses that come through after the various treatments are merely plant viruses. They're not animal viruses. So the water then, we can demonstrate, is very safe. One question that was really critical was antibiotic resistance being transferred to the water bacteria from the pathogens that came in the sewage water? And that was a real issue, but we were able to show, because we can demonstrate the presence or absence of the antibiotic resistance genes, that by the time it goes through the reverse osmosis, there are very, very few antibiotic resistance genes, so that does not occur, and so that is a safety factor. The last point is how do you bring all this technology of satellites and sensors and DNA sequencing and all of this and help the villagers in the remote parts of Bangladesh and India? And that is understanding this relationship, knowing that these little critters are pretty big, they're the elephants of the microscopic world, and folding sari cloth and filtering the water gives you a 20 micrometer mesh. The bacteria are 2, 5, at the most 10 micrometers. A few slip through if you filter, but most don't. You can remove 99% of the bacteria. The water is looks a whole lot better than water from the pond with things swimming in it. And in fact, we could reduce cholera 50% using the science evidence to provide a simple solution. So I conclude with the words of John Muir: when one tugs at a single thing in nature, he and she finds it hitched to the rest of the universe. And a lot of people have been involved in the work in Bangladesh, in Calcutta, colleagues, students, technicians, postdocs, visiting scientists from other countries. And I'd like to thank those who worked with me on some of the developing the bioforensic work, and special thanks to 4 people who have helped so much. Dr. Huck, who has worked with me, he's now a professor at the university for the last 20-some years. Antar, who continues to work with the satellite, and Hassan, who does so much of the elegant informatics and DNA work, and Sion Young, who is a total gem on curating all those bacteria and giving us perfectly clean sequences of all the strains, in the genomics team. And the main point of the work is to move us away from this situation so that we can help these people have access to safe water. Thank you.
Some time for questions if people want to hang out for a little bit and ask. We have live mics here, so please take advantage.
I've taken through 40 years' work in 40 minutes. How's that? 1 year a minute. No questions.
Thank you, Rita. That was amazing. I had a question about the development of the copepod or other plankton as a vector, and then the connection to humans. Do you think this was an evolutionary adaptation between the 3, or just by chance hanging out on chitin and sort of keeps them?
I think that's a great question, Colleen, because the receptor sites in the human gut have been shown to be somewhat similar, strangely enough. Or maybe not, because if you believe in evolution, it should be rational. to some of the sites in the gill and the tissue in the invertebrates. But more to the point, I think that the relationship is such that the toxin that's produced by Vibrio cholerae is probably an osmoregulator for the host, because crabs migrate from seawater to the freshwater of the upper bay, and these copepods, many of them do much the same, and it is not really known how they osmoregulate. And so we know that in humans it affects the sodium-potassium transport and causes an efflux of fluid, which causes the dehydration. And if you put it in sort of the vernacular, the bacteria may think we're just one great big copepod. And hence, it's not an evolutionary relationship, but it's inadvertent host relationship. And I think that's probably more likely.
Where are the bacteria localized? I thought that they were
On the-
colonizing the chitin, but
It's on the gills
so the gills.
and the exi, where the appendages are attached, the cluster. And then as the copepod ages, then you find it on the chitin surface. We tried to do ligated crab loops at Woods Hole with radiolabeled sodium, not a great idea. And ligating a rabbit loop is one thing. Ligating a crab loop is quite another. And the experiment didn't work because we were hoping we could show osmoregulation. Shows you extreme scientists will go to prove a point. Yes.
Do you think with global warming that cholera is going to become endemic in more parts of the world, and can you predict that based upon, I guess, your satellite technology?
Yes, the question about cholera becoming more dominant, I don't think in Western world, with the exception that I would make that with the increasing frequency of very severe storms and winds that will break down water treatment plants and create sewage treatment plant breakdown as occurred. I think it was Milwaukee where the cryptosporidium outbreak occurred. That sort of situation could cause outbreaks. We were very fortunate in Louisiana when the hurricanes occurred. We did not have outbreaks of cholera. We did have a few cases, but bottled water was shipped in, so we avoided that. I think for the developing countries, it'll be a problem, but I think for Bangladesh, the bigger problem is that 20% at least of the country will be flooded within the next 30 to 50 years with sea level rise. So I think the problem we're going to have is probably 20 or 30 million climate change refugees, and that's going to be a big problem. Yes?
Thank you, Rita. Are there, going back to the copepods, are there other bacteria on the copepods that do play a role in the Vibrio biogenesis? And secondly, there's a lot of biodiversity, within copepods. So how restricted is the bacterium within copepods?
Well, first of all, the copepod, freshwater copepods carry Vibrios. And that explains why, for example, in the Lewis and Clark expedition, some of them got cholera as they went west. And one could hypothesize, I'm allowed to hypothesize, so danger, that with glaciation and receding, that you may have ended up with the copepods then as part of what was left of the receding glacial waters, and the copepods then remained entrained in the lakes. And also I've isolated Vibrio, not cholera, but Vibrio species easily from the dry areas of Antarctica. Now that may come in with winds, or that may be a residual from prehistoric times. It doesn't seem to matter so much about different species of copepods. It's the chitin exoskeleton that's the critical aspect of it. And some of the phytoplankton that do carry bits of chitin in their structure may have some Vibrios as well, but the dominance is by far the greatest. And the copepods represent a dominant portion, particularly, for example, ballast water of ships is almost 90% copepods, and therefore that is one of the reasons when ballast is dumped, you can have an outbreak.
So is there something specific to the chitin in copepods as opposed to other arthropods? Or is it the life history of copepods that puts the bacterium in the right place to cause it?
It's a life history. I don't think it's anything special about the chitin. We've done a lot of studies using chitin that you get commercially to show chitin digestion. Yes?
I'm just wondering, a lot of the regions with cholera outbreaks are limestone regions with big karst aquifers underneath them. And there's, I happen to study the karst aquifers, so I'm interested in that. Have you, by chance, sampled these groundwater environments and the crustacean-dominated communities that live within them? There's a lot of copepods.
I would love to do that. I'll talk to you later. We'll set up an experiment. It makes great sense. It makes great sense. And we've actually isolated one of my students, Brad Haley, did a Fulbright in Iceland and isolated Vibrio cholerae from where the heated geothermal waters temperatures were very high. So they're globally distributed. They're part of the natural flora of the temperate and warm water areas. So that would be a normal thing to look at. And yes, I would expect Vibrios. Would we necessarily expect Vibrio cholerae? It would depend on whether in those areas there were copepods as well, or crustaceans.
Oh, there are. It would be interesting. Also, a lot of those regions have the entrances to the groundwater environments are used as latrines and bathing sources.
It'd be very interesting. Yes.
About 70 years ago, J.B.S. Haldane wrote a great paper on disease and evolution. In this, he predicted that diseases could be like cholera and others could be great selective agents. If so, in your work, great body of work, have you seen differential effects of cholera on different host populations?
Yes, you're going to tax my memory, though. One of the reasons that was offered for the devastation of the Latin American outbreak was because of the difference in the blood type. And it turns out there's a susceptibility of one blood type to cholera. And so there is a selective process that goes on. And I think, having read some of Haldane's papers, I think that's probably the basis of a blood type genetic phenomenon. But that's a very interesting question. Yes?
I was just curious as why the nylon was less effective than the sari. Is it just a bigger mesh, or?
It's a slightly larger mesh, and also it's more expensive too. The nylon, we use that because it has been successful in dracunculiasis, guinea worm in Africa, where there's an intermediate stage in copepods. And it was introduced to provide separation of the copepods, which carry that larval stage for that infection. We used it simply because it had been used and was available, but it's almost a month's wages for a villager to have a yard of the nylon mesh. And the used sari cloth, everybody has it available at no cost. Yes?
I was curious again about the link between, say, climate change and potential outbreak areas of cholera along coastal areas, because two things are happening at once, several, but increased temperature is one, but sea level rise is another.
Yes.
And if you look particularly at the post-Columbian cities, the cities put in place since Columbus, like all of the capital cities of West Africa, for example, places like Nigeria are going to be inundated, cities of 6 to 8 million in the next 30 years. They already have sort of limited waste evacuation infrastructures. Can we expect these epidemics to be flourishing in these areas?
Well, there'll be a couple of factors at play. You will have greater crowding because the tendency is to move to the big cities, and we already have mega cities. And then if you bring individuals that come from a poor sanitation area, perhaps carrying, not necessarily cholera, but there's 26 diseases. By the way, you're giving me an opportunity to make one point I really wanted to make. If you provide safe water and sanitation, you deal immediately with 26 diseases transmitted by water, for which only 2 are there vaccines. Rotavirus, which is very expensive and requires a cold chain, and cholera, which is not, at best, 70%, but more likely 60% effective, and usually for 2 to maybe 7 years. But if you provide safe water, how many of us in this room have had cholera vaccine? Well, only maybe a few of us who went to Bangladesh. And when I went to Bangladesh several years ago, they used to require it, and my good friend said, "The vaccine's not so hard, I'll just sign that you've taken it so you don't have to take it." And I shouldn't have said that, I'm being recorded. But the point is that you can really deal with human health in a fantastic way by providing safe water and sanitation. But back to your point, with sea level rise, we will have a public health problem. Any other, on that happy note, thank you all, it's been a pleasure.